good morning, good afternoon. Have to check my watch to know what time it is. Good morning. I would like to introduce Brittany White. Brittany White is a graduate student in the biology department. She's working on her thesis project. Her thesis entails, actually she picked up and collaborated with an undergrad who started the project, Trinity, Elston, who's presented here in the past. Brittany's picking up and actually doing a little bit of more bioinformatics research and examining the impact of a potential patient mutation in the gene INSR, which is the insulin-like receptor. So Brittany, without further ado, I will let you take it off.
And judges, if you need to step up, you're welcome to get up closer to the school.
Thank you.
So we are looking at a variant of unknown significance, or it's known a VUS. So in this region, we're looking at some characteristics of type 2 diabetes. So we are looking at insulin resistance. So this gene, there are several known mutations that are pathogenic, so they do contribute to type 2 diabetes. But this one, it's unknown if it does cause it or not. So we do have, again, it does harbor these genetic variants. Let me move this way so y'all can see a little bit better. So we are looking at the amino acid 543, which in a wild type or a normal, it would be threonine. And then with the mutation, we are going to methionine. So there is a class change in the amino acid. And so looking at that class change, we are predicting, our hypothesis would be that this class change and the structural change in this protein would affect the functionality of that protein. So looking at materials and methods, we did use several bioinformatics softwares. So very first thing, Dr. Turner was a great support in providing some protocols. So we did use those protocols both in the lab setting and as well with the bioinformatics softwares. Benchling was the most prominent bioinformatics tool that I've used. It was used to create RNA guides, to create primers, and it was also used to construct the MSA that we'll talk about here in a little bit, as well as to confirm conservation, to locate these amino acids in both the nematode and the human gene. We did use Microsoft Teams and SharePoint as a way to help keep track of the electronic lab notebook, as well as to be able to communicate back and forth. OneNote was also used for that purpose as well with the ELN. Uniprot gave us the information to be able to construct the models in Swiss model. So that would be moving into Swiss model there. Swiss model allowed us to generate the PDB files to then put into PyMol. So creating the protein models that we'll talk about took several different softwares to be able to generate those. And then PyMol allowed us to compare the wild type and the variant. And then with all of this, you do need working internet and a computer for that. And we also did some PCR. So this is still ongoing. So we did do some PCR and gel electrophoresis analysis. So we do have primers in the lab that are currently being used on this project. So looking at what we have so far, this right here is our MSA, or our multiple sequence alignment. So we are looking at conservation across multiple species. And looking at research from other articles, the insulin signaling pathway is one of the most conserved pathways across several species. So there's plenty of information to be able to see that, and we see that it is conserved between the Homo sapiens, humans, and C. elegans, the nematode model that we are using. So our MSA definitely focused on those two. So when we're looking at the C. elegans, what we have is DAF2. It is the ortholog of INSR in humans, and there is known phenotypes about it, which we'll bring that point up again. This right here is a polyphen-2 analysis. When you do this analysis, there are two values it gives you. You can have either a HumDiv or a HumVar. HumDiv is for Mendelian diseases. Type 2 diabetes is not that. It's a little bit messier. So we did go with the HumVar, and it is predicted to be possibly damaging. So this is a tool to predict pathogenicity. With this high of a score, it predicted at 0.954. this definitely indicates that this is a project that we wanted to move forward with. And then looking right here at figure 3, these are our protein models. So in green, we have our wild type, and then in the teal, we have our variants. And then looking right here, we have it superimposed. So they are overlaid on top of each other here. So when we're looking at this amino acid change, again, I did mention that there is a class change. So we are going from non-polar to polar. Further research has shown that this happens in the extracellular matrix. So the INSR gene is a tetramer, and this is going to be in that alpha unit in the extracellular space. So this could potentially be why we would have such a high prediction for pathogenicity, even though it is not in a higher order structure. It is not in an alpha sheet or a beta helix. It is in a glycosylation. But it could end up impacting how insulin would bind with the cell itself. So looking at our discussion, we do have a class change from polar to non-polar there. And this does cause a structural change, as you can see right here. And it was a noted structural change with the RMSD value of 0.001. That doesn't seem like much, but again, could potentially impact how insulin is binding. And then we did just talk about the extracellular alpha subunit of the INSR gene being extracellular, again, insulin binding. And then, because we can predict these things, this does warrant further investigation. So next steps for this would be we've already completed some PCR, so we do have primers that are working. We have tested them and optimized them. We do have RNA guides designed, so next step is to purchase those, do in vitro assays, and then look at phenotyping. We will be using CRISPR to generate a DAF2 mutant in the C. elegans to be able to test this. Since it is an ortholog of the human INSR, it could give us some insight into how INSR is being affected by this mutation. So again, we're looking at a potential to be able to find yet another pathogenic variant for type 2 diabetes here. So this can definitely contribute into our knowledge of type 2 diabetes. And I would like to thank Dr. Turner and of course the biology department. They've been very supportive. So what questions would we have about this?
I had a question. Do nematodes make insulin?
So they do not make insulin, but the DAF2 signaling pathway is very conserved along with the insulin signaling pathway. They are orthologs of each other. Though it may be different players in the game is still the same. That's a good analogy for that.
So you mentioned that you focus primarily on human and nematodes. Were there other organisms? And if so, what were they?
So we do have other organisms up here, such as mouse, zebrafish. It is conserved with them, but we focus on C. elegans just because that's the model we're using in the lab.
Thank you, Judge, first.
Okay. This is not something I know very much about, so I want to see if I understand what you've done here. Your images that you have there showing the change from the wild type to the non-wild type, that's based on computer modeling?
Yes, in Python.
Okay, all right. So the next step then is to actually use CRISPR and see what happens in the actual images.
Yes, and observe those phenotypes. And there are known phenotypes, so with DAF2, if there is a mutation in it, or if that gene is not functioning correctly, we would have things such as Dower formation, which is a stress response for the C. elegans, and as well as an extended lifespan, mainly because of that Dower formation. It's pretty much a larval arrest. So when they form Dowers, then the development is arrested for a period of time.
So this change would cause arresting?
So this right here changed because of amino acid class change. So the way the amino acid is interacting with other amino acids surrounding it, we do have our class change here. It changes its behavior just slightly. This one would be our polar, and then our non-polar right here. So one is going to be hydrophilic and one hydrophobic. Just the way it interacts with water and
That's kind of where I was actually going. So, my question was, you mentioned that this is in the extracellular site on the receptor. Does this protein have the receptor site for insulin mapped, so you know exactly where that is?
so it is not fully mapped. There is some. Now, I haven't had a chance to look at other bioinformatics sites yet, but in Uniprot, part of this was missing, especially when we were looking at comparing it to DAF2. There is some of the sequence that is missing.
And the only reason I asked was because you said this was not in an alpha helix, not in a beta helix structure. So I'm assuming it's in kind of a bend area, turn area, something like that in that structure.
Yeah, it is in a loop.
It's in a loop. It's kind of what was left in my mind. So I was just wondering, because you were talking about it's obviously going to change because it's changing that interaction. I didn't know if it was a direct receptor site or an indirect relationship on the receptor site. We don't know, so that's the answer.
Yeah, there is definitely still ongoing research on INSR, not just from here, but other places. So hopefully in the next coming years, we will definitely have some more information about this.
And INSR has already been crystallized, it's just the DAF2 nematode ortholog has not been crystallized. So there's a lot more known about the INSR molecule than there is the DAF2 molecule.
And I know nothing about nematodes or DAF2, so I'm sorry to go here.
No.
Is it related to energy function in?
Oh, 100%. So
Okay.
this is the cell sensing nutrients, this is the nutrient sensing cycle or pathway. Yep.
Okay, any last questions for our speaker? All right, well, let's thank them again.